Hello and welcome dear students. Today we are going to discuss about the topic genotype phenotype correlations. The main objectives of today's lecture are to know about the basics of genotype and phenotype, to study the factors affecting genotype and phenotype, to know about the relation between genotype and phenotype, and finally to elaborate view of the correlation between genotypes and phenotypes in terms of various laws. Dear students, let's first start with the introduction of genotype and phenotypes. Genetic code is responsible for your traits or characteristics. Your genotype is your genetic code of your cells. Genetic code refers to the instructions contained in the gene that tells a cell how to make a specific protein. A gene in turn is a portion of DNA that codes for a particular trait, and each gene's code uses the 4 nucleotide bases of the DNA, that is the adenine, guanine, cytosine and thymine, in various ways to spell out 3-letter code, that is the codons, that specify which amino acid is needed at each position within a protein. Your genetic information is stored in your chromosomes on sections called genes. So genotype can be described as the genetic makeup of an organism. The physical expression of your genotype, that is the genotype has been derived from the Greek genos, meaning race or offspring, is your phenotype, that is the from Greek pheno and ultimately from the phenein, meaning to show. As humans are diploid organisms that have 2 alleles at each genetic position, with 1 allele inherited from each parent, male and female. Phenotype refers to the physical properties of an organism which can be observed with our eyes. These include organism's appearance, development, behavior, height, color of eyes, hairs, etc. The concept of genotype and phenotype was introduced by Janssen in 1909 in his textbook on heredity research. He further developed this concept in a paper named the Genotype Conception of Heredity in 1911. An organism's genotype affects its phenotype, and the change in the environment also affects phenotype. So the genotype-phenotype distinction is drawn in genetics. Genotype is an organism's full hereditary information, while as the phenotype is an organism's actual observed properties such as morphology, development or behavior. This distinction is fundamental in the study of inheritance of traits and their evolution. The genotype is a set of genes in the DNA which are responsible for the unique trait or characteristic to make your phenotype, whereas the phenotype is the physical appearance or characteristics of the organism. Thus, we can find the human genetic code with the help of their genotype. Organisms may look the same but still may not have the same genotype. One can determine the genotype by the biological tests. On the other hand, the phenotype is determined by an individual's genotype and can be expressed as genes or by the visible traits such as hair color, eye color, body shape, height, etc. In biology, a gene is a part of the DNA that encodes some traits. The precise arrangement of nucleotides, each composed of a phosphate group such as sugar and a base in a gene, can differ between copies of the same gene. Thus, a gene can exist in various forms across organisms. These different forms are popularly known as alleles or alternative forms of the gene. Also, the exact as well as a fixed position on the chromosome containing a particular gene is termed as the locus. A diploid organism may inherit 2 copies of the same allele or may inherit 1 copy of 2 different alleles from their parents. So, the individual inheriting two identical alleles is termed as homozygous at that particular locus. Whereas, if they possess two different alleles, then their genotype will be classed as heterozygous for that locus. Alleles having the same gene are either autosomal dominant or autosomal recessive. The subsequent combination of alleles that are possessed by the individual for a specific gene is their genotype. Phenotype, on the other hand, refers to an individual's observable traits such as height, eye color, and blood type. A person's phenotype is determined by both their genomic makeup, that is the genotype, and the environmental factors. In genetics, the phenotype is the set of observable characteristics or traits of an organism. The term covers the organism's morphology or physiological form and structure, its developmental processes, its biochemical and physiological properties, its behavior, and the products of behavior. A key difference between phenotype and genotype is that while genotype is completely inherited from the parents, but the phenotype is not, as because the phenotype is influenced not by the genotype only, but by many other factors like epigenetic modifications and environmental and lifestyle factors. Environmental factors that are influencing the phenotype include nutrition, temperature, humidity, and stress. Flamingos are a suitable example of how the environment influences the phenotype, as they are renowned for being vibrantly pink, but their natural color is white, and the pink color is caused by pigments in the organism in their diet. Our genes can control the amount and type of melanin produced by us, but exposure to UV light in sunny climates causes the darkening of existing melanin, and hence it causes darker skin. Dear students, it was all about the phenotype and genotype definitions. Let's now discuss the factors affecting genotype and phenotype. The genotype of a person can be affected by the environment in which the person lives. Environmental factors such as diet, temperature, oxygen levels, humidity, light cycles, and the presence of mutagens can all impact which of an animal's genes are expressed, which ultimately affects the animal's phenotype. The genotype of a person can also be affected by the internal environment of a person's body, such as presence of hormones and metabolism. Although phenotype is affected by the genotype of a person, but it has been observed that even genetically identical organisms exposed to controlled experimental conditions can have different phenotypes, pointing to the power of subtle environmental differences on gene expression. Now, what are the types of genotypes? A combination of alleles situated on corresponding chromosome determines a person's specific traits. The genes responsible for traits such as color of the skin can be example of a genotype. There are three kinds of genotypes based on alleles of a person. It can be homozygous dominant, it is represented by capital letters P and P. Heterozygous, when it is represented by a capital letter P and a small p. And homozygous recessive, when it is represented by small p, small p. So, homozygous and heterozygous are terms that are used to describe allele pairs. Being homozygous for a particular gene means you inherited two identical versions. It's the opposite of a heterozygous genotype where the alleles are different. People who have recessive traits like blue eyes or red hair are always homozygous for that gene. The genotype having more dominant genes is considered as strongest because dark hair, right-handedness, brown eyes, left thumb crossing, long eyelashes, left arm over right, broad lips, etc. In hot climate regions, certain races have broad lips as their dominant gene. Dear students, let's now discuss the genotype and phenotype correlations. One of Gregor Mendel's greatest contributions to the study of heredity was the concept of dominance. Mendel observed that a heterozygote offspring can show the same phenotype as the parental homozygote, so he concluded that there were some traits that dominated over other inherited traits from the parents. However, the relationship of genotype to phenotype is rarely as simple as the dominant and recessive patterns described by the Mendel. As the study of inheritance expanded beyond Mendel, Mendel initially examined and also included organisms other than pea plants. Biologists began to notice a variety of relationships between alleles that code for the same trait. These allelic interactions were not exclusively recessive or dominant, and they greatly enriched our understanding of how genotype leads to phenotype. Dear students, let's now discuss the complete versus partial dominance. Dominance affects the phenotype derived from an organism's genes, but it does not affect the way these genes are inherited. Complete dominance occurs when the heterozygote phenotype is indistinguishable from that of the homozygous parent. However, sometimes the heterozygote displays a phenotype that is an intermediate between the phenotypes of both homozygote parents, one of which is homozygous dominant and the other of which is homozygous recessive. This intermediate phenotype is a demonstration of partial or incomplete dominance. When partial dominance occurs, a range of phenotypes is usually observed among the offsprings. Note, however, that partial dominance is not the same as blending inheritance. After all, when two F1 generation pink flowers are crossed, both red and white flowers are found among the progeny. In other words, nothing is different about the way these alleles are inherited from parents to offsprings. The only difference in this way is the allele determine the phenotype when they are combined. Let's discuss the codominance. Codominance, as opposed to partial dominance, codominance occurs when the phenotypes of both parents are simultaneously expressed in the same offspring. Indeed, codominance is the specific term for a system in which an allele from each homozygote parent combines in the offspring and the offspring simultaneously demonstrates both phenotypes. An example of codominance occurs in the human ABO blood group system. Many blood proteins contribute to blood type, and the ABO protein system in particular defines which types of blood you can receive in a transfusion. In a hospital setting, attention to the blood proteins present in an individual's blood cells can make the difference between improving health and causing severe illness. There are 3 common alleles in the ABO system. These alleles segregate and assort into 6 genotypes. Only 4 phenotypes result from the 6 possible ABO genotypes. How does this happen? To understand this, why this occurs, first note that the A and B alleles code for proteins that exist on the surface of red blood cells. Thus, if one parent is homozygous for type A blood and the other is homozygous for type B blood, the offspring will have a new phenotype, that is type AB. So, in people with type AB blood, both A and B proteins are expressed on the surface of the RBCs or red blood cells equally. Therefore, this AB phenotype is not an intermediate of the 2 parental phenotypes, but rather is an entirely new phenotype that results from codominance of the A and B alleles. Of course, in this system, AB is not the only heterozygote. People may also be AO or BO. All of these heterozygote genotypes demonstrate the coexistence of 2 phenotypes within the same individual. Dear students, let's now talk about the overdominance. In some instances, offsprings can demonstrate a phenotype that is outside the range defined by both parents. In particular, the phenomenon known as overdominance occurs when a heterozygote has a more extreme phenotype than that of either of its parents. Indeed, in a few examples, a trait that shows overdominance sometimes confers a survival advantage in the heterozygote. A well-known example of overdominance occurs in the alleles that code for sickle cell anemia. Sickle cell anemia is a debilitating disease of the red blood cells or RBCs wherein a single amino acid deletion causes a change in the conformation of a person's hemoglobin such that person's red blood cells are elongated and somewhat curved, taking on a sickle shape. This change in shape makes the sickle red blood cells less efficient at transporting the oxygen through the bloodstream. The altered form of hemoglobin that causes sickle cell anemia is inherited as a codominant trait. Specifically, heterozygous, that is capital S and small s individuals, express both normal and sickle hemoglobin. So they have a mixture of normal and sickle red blood cells. In most situations, individuals who are heterozygous for sickle cell anemia are phenotypically normal. Under these circumstances, sickle cell disease is a recessive trait. Individuals who are homozygous for the sickle cell allele, that is small s or recessive, however, may have sickling crises that require hospitalization. In severe cases, this condition can be lethal. Producing altered hemoglobin can be beneficial for inhabitants of countries afflicted with falciparum malaria, an extremely deadly parasitic disease. So sickle blood cells collapse around the parasites and filter them out of the blood. Thus, people who carry the sickle cell allele are more likely to recover from malarial infection. In terms of combating malaria, the heterozygote, that is capital S small s genotype, has an advantage over both, that is the homozygote dominant capital S genotype, because it results in malarial resistance, and the small s, that is the recessive homozygote genotype, because it does not cause sickling crisis. This complex example of overdominance may be the sole reason that the allele persists in the human population even today. Dear students, let's now discuss the multiple alleles and the dominance series. Allelic dominance always depends on the relative influence of each allele for a specific phenotype. Under certain environmental conditions, multiple alleles exist at the population level, and an individual can have only 2 alleles for the given gene. An individual can have only one type of allele, that is homozygous, or different alleles, that is heterozygous. A series of mutant alleles can exist in a population. So dominance is a relationship between 2 alleles of one gene that affect the phenotype of one allele and masks the contribution of another allele. So the trait which is expressed in a phenotype is called the dominant trait, and the suppressed one is called a recessive trait. In genetics, dominance is the phenomena of one variant allele of a gene on a chromosome masking or overriding the effect of a different variant of the same gene on the other copy of the chromosome. The first variant is termed dominant and the second is termed as recessive. Dear students, summarizing the role of dominance and recessivity. Mendel's early work with pea plants provided the foundational knowledge for genetics, but Mendel's simple example of 2 alleles, one dominant and one recessive for a given gene, is a rarity now. In fact, dominance and recessiveness are not actually allelic properties. Rather, they are effects that can only be measured in relation to the effects of other alleles at the same locus. Furthermore, dominance may change according to the level of organization of the phenotype. Variations of dominance highlight the complexity of understanding genetic influences on phenotypes. Mendel's hybridization experiments demonstrate the difference between phenotype and genotype. For example, the phenotypes that Mendel observed in his crosses between pea plants with different traits are connected to the diploid genotypes of the plants in the pea, that is parental, F1 and F2 generations. We will use a second trait that Mendel investigated, that is the seed color, as an example. Seed color is governed by a single gene with two alleles. The yellow seed allele is dominant and the green seed allele is recessive. When true breeding plants were cross-fertilized in which one parent had yellow seeds and the other had green seeds, all of the F1 hybrid offsprings had yellow seeds. That is, the hybrid offspring were phenotypically identical to the true breeding parent with yellow seeds. However, we know that the allele donated by the parent with green seeds was not simply lost because it reappeared in some of the F2 offsprings. Therefore, the F1 plants must have been genotypically different from the parent with yellow seeds. Dear students, let's now discuss the law of dominance. Our discussion of homozygous and heterozygous organisms brings us to why the F1 heterozygous offsprings were identical to one of the parents rather than expressing both alleles. In all 7 pea plant characteristics, one of the two contrasting alleles was dominant and the other was recessive. Mendel called the dominant allele the expressed unit factor. The recessive allele was referred to as the latent unit factor. We now know that these so-called unit factors are actually genes on homologous chromosomes. For a gene that is expressed in a dominant and recessive pattern, homozygous dominant and heterozygous organisms will look identical. That is, they will have different genotypes but the same phenotype. And the recessive allele will only be observed in homozygous recessive individuals. Dear students, let's now discuss the law of segregation. Observing that true breeding pea plants with contrasting traits give rise to F1 generations that all express the dominant trait and F2 generations that express the dominant and recessive traits in a 3:1 ratio, Mendel proposed the law of segregation. This law states that such offsprings have an equal likelihood of inheriting either factor. For the F2 generation of a monohybrid cross, the following 3 possible combinations of genotypes result, that is homozygous dominant, heterozygous or homozygous recessive. Because heterozygotes could arise from 2 different pathways receiving one dominant and one recessive allele from either parent, and because heterozygotes and homozygous dominant individuals are phenotypically identical, the law supports Mendel's observed 3:1 phenotypic ratio. The equal segregation of alleles is the reason we can apply the Punnett-Square to accurately predict the offsprings of parents with known genotypes. The physical basis of Mendel's law of segregation is the first division of meiosis, in which the homologous chromosomes with their different versions of each gene are segregated into daughter nuclei. This process was not understood by the scientific community during Mendel's lifetime. Dear students, let's now discuss Mendel's law of independent assortment. So this law states that and every possible combination of alleles for every gene is equally likely to occur. Independent assortment of genes can be illustrated by the dihybrid cross, a cross between 2 true breeding parents that express different traits for 2 characteristics. The physical basis for the law of independent assortment also lies in meiosis 1, in which the different homologous pairs line up in random orientations. Each gamete can contain any combination of paternal and maternal chromosomes and therefore the genes on them, because the orientation of tetrads that is the 4 homologous chromosome arms on the metaphase plane, is random. Dear students, let us now discuss the correlation between genotype and phenotype in a nutshell. The correlation between genotype and phenotype is a statistical relationship that predicts a physical trait in a person or abnormality in a patient with a given mutation or a group of similar mutations. The correlation between genotype and phenotype is defined as an above chance probability of a distinct mutation being associated with a particular physical feature or abnormality. The more frequently a specific phenotype is observed in association with a certain genotype, the higher the likelihood that an unrelated person with the same genotype will show the traits or abnormalities observed in the population carrying the same alleles. Linking genotype to phenotype is a major aim of genetics research, yet the underlying biochemical mechanisms of many complex conditions continue to remain elusive. Phenomics is a new and emerging area of study underlying the development of genotype-phenotype mapping and the identification of different disease interaction networks. Genotype-phenotype mapping involves the delineation of a relationship between the genetic constitution of an individual and an observable set of characteristics of interest. While the relationship between a phenotype and a given genotype is complex, in the case of rare disorders with high penetrance mutations, determining such a relationship becomes relatively easier, although may still require the aid of computational methods. Dear students, it was all about today's lecture on phenotype-genotype relationship and their correlation. Hope you have understood it well. Take care. Goodbye.